Sexual reproduction in flowering plants. Flowering plants reproduce sexually through a process called pollination. Plants can either self-pollinate or cross-pollinate. Self-pollination happens when a plant's own pollen fertilizes its own ovules. Cross-pollination, wind or animals move pollen from one plant to fertilize the ovules on a different plant. Pollinators are animals that carry pollen between plants. Many pollinators are insects, like bees, butterflies, moths and beetles. Some birds, including hummingbirds, part. Likewise, certain mammals, like bats and rodents, move pollen between plants. Flower structure. The flowers contain male sex organs called stamens and female sex organs called pistils. The anther is the part of the stamen that contains pollen. This pollen needs to be moved to a part of the pistil called the stigma. There are two parts of a typical, the long and slender stalk called the filament, and the terminal generally bilobed structure called the anther. The microsporangia develop further and become pollen sacs. They extend longitudinally all through the length of an anther and are packed with pollen grains. When the pollen grain is matured, it contains the vegetative cell and generative cell. Development of male gametophyte. The pollen grains represent the male gametophytes. Male gametogenesis begins with the division of a diploid sporophytic cell, giving rise to the tapetal initial and the sporogenous initial, that is the pollen mother cell. The sporogenous cells undergo giving rise to a tetrad of haploid cells. The individual cells of the tetrad are released as free microspores by the action of an enzyme callus. These uninucleate microspores undergo an asymmetric mitotic division, resulting in a pollen grain containing two cells, a larger smaller generative cell that is enclosed entirely within the vegetative cell. A second mitotic division occurs before the pollen is shed from the plant. When once they are shed, pollen grains have to land on the stigma before they lose viability if they have to bring about fertilization. Development of female gametophyte. The gynoecium represents the female reproductive part of the flower. The gynoecium may consist of a single pistil, or monocarpellary, or may have more than one pistil, or multicarpellary. Each pistil has three parts: the stigma, style and the stigma serves as a landing platform for pollen grains. The style is the elongated slender part beneath the stigma. The basal bulged part of the pistil is the ovary. Inside the ovary is the ovarian cavity, or locule. The placenta is located inside the ovary. Arising from the placenta are the megasporangia, commonly called ovules. Each ovule has one or two protective envelopes called integuments. Integuments encircle the nucellus except at the tip where a small opening called the micropyle is organized. Enclosed within the integuments is called the nucellus. Located in the nucellus is the embryo sac or female gametophyte. An ovule generally has a single embryo sac formed from a megaspore. The process of formation of megaspores from the megaspore mother cell is called megasporogenesis. In flowering plants, one of the megaspores is functional while the other three degenerate. Only the functional megaspore develops into the female gametophyte. The nucleus of the functional megaspore divides mitotically to form two nuclei which move to the opposite poles, forming the two nucleate. Two more sequential mitotic nuclear divisions result in the formation of the four nucleate and later the eight nucleate stages of the embryo sac.